my PhD from State University of New York in Stony Brook.
Okay.
And then my postdoc at MIT and Harvard.
Okay. And
Currently, I'm in Indian Institute of Public Health, Hyderabad. I'll introduce myself.
all right. So, welcome to the final day of the workshop. And as a speaker, the first speaker, we have Professor Soumyadipta Pyne. Professor Pyne did his PhD at State University of New York, Stony Brook, and then did postdocs at MIT and Harvard. Yeah, and he's currently at the Institute of Public Health at Hyderabad. So, Professor Pyne.
Hello. Okay. A very good morning to all. So firstly, thanks to the organizers for inviting me and giving me the opportunity to present the work that we do. So today's first talk, as a part of the workshop talk, is probably less going to be the work that we directly do at this point of time, more in the flavor of general kind of work that's done by the community in this particular area, because I kind of figured today maybe a workshop audience, which is generally comprised more of students, and they would be benefited more in terms of the general kind of work that's done in the area rather than a very specific research projects or research-oriented problems that we do in our labs. So which will be covered in my tomorrow's talk. Those will be kind of talks about the very precise problems that we cover in the lab rather than the general kind of theme that I'll expose you to today. So today is more like a prelude to tomorrow's work or tomorrow's presentation. Okay. So a little bit of the background. I had a few slides about the institutes which I had taken out because I thought maybe I would have less time. So I'll just give you the background orally. So I'm now located in a city called Hyderabad, which is in the neighboring state just north of Tamil Nadu, for those who are not familiar with that. So and this institute is called Indian Institute of Public Health, which is one of the 5 institutes of the Public Health Foundation of India. So Public Health Foundation of India was founded with the help of the Prime Minister's office and the Ministry of Health in 2006, I think. And then the first institute that the foundation created was in Hyderabad. It's called Indian Institute of Public Health of Hyderabad. And now there are 5 Indian Institutes of Public Health in 5 zones of the country, including one in the Northeast. And of course, more are on the way. So we have about 70 plus faculty all over the country and 26 in Hyderabad. Okay, so that's where Indian Institute of Hyderabad stands. And you see the other logo here, because that's my other remote affiliation with Broad Institute of MIT and Harvard University. So our team and colleagues over there work on certain aspects which I'll touch upon tomorrow, not today. So that's a good amount of our data come from there. And along with that, so that's where our lab has been working for almost a decade now. But so when I planned to return to India about 2.5 years ago, almost 3 years ago, so where I came back to, and that institute is not listed here, and that's what I was saying, the slides I had taken out, is an institute called CRO, Advanced Institute of Mathematics, Statistics, and Computer Science. That institute is also located in Hyderabad, and more so within in the campus of University of Hyderabad. Okay, so that institute still exists. I just recently relocated, but that institute is focused, the area that I was developing there, and I still do some work over there, is biosecurity and the kind of work that I'll describe today. Okay, they have other wings like cybersecurity and so on. Okay, so sorry about the date. Okay, that's the wrong date. Okay, so these are the research directions of this kind of Indo-US type lab. So it's kind of two directions, one in bioinformatics, other in biostatistics. And the bioinformatics dimension is basically comprises of platforms that we created since the Human Genome Project ended. So it's more than a decade's work. So about 13, 14 years worth of work. So most of these are computational statistics platforms that we created in Broad Institute of MIT to support the large amounts of data that were generated following the Human Genome Project. The different omics platforms were flourishing, and they were kind of spitting out new data sets for which people didn't know how to make sense of. Because suddenly, if you're coming from a background like I did from statistical genetics, where we looked at few genes and kind of made very rigorous hypothesis testing there, those type of approaches were not very feasible. Suddenly, around I would say 2002 to 5 in that era. Suddenly there were these genomic platforms, large transcriptomic platforms, and all other omic platforms. So they were generating data on tens of thousands of variables per subject. And suddenly we had all these kinds of statistical challenges, not just in inference, but also most importantly, we had machine learning challenges to even validate whether that data even is real or just some arbitrary bunch of numbers. Okay, so we had to validate all that, and then we had to come up with suites of tests and analytical tools to help others, the people who applied such data for their research, and to support them and make sure that we could collaborate. And then the Harvard Medical School teams and other teams in both in Europe and United States started using these. And so now these are a bunch of very popular platforms that we had created, and now a lot of users around the world. Then around 2005 to 6, we started kind of changing focus from high throughput data to high resolution data. The difference is in high throughput data, you measure a large number of variables, say tens of thousands of genes, right, per person, per sample or subject. But in high resolution data, in addition to the high throughput nature, there's also this other technological aspect that we had to understand. And that's when you're looking at measurements at single cell level, or even finer than that. Okay? So now, for example, microarray is a gene expression microarray is a common type of data that's generated from the microarray platform, which looks at thousands of genes. Yes, true, that's high throughput, but it doesn't look at single cell resolution, nowhere close to that. It takes a sample which is, let's say, millions of cells. So when it says that this particular gene is expressed in this sample, then it says that as an average of what it could measure over those millions of cells in that sample. But so it's not really high resolution. So now we said that we would be happy to develop platforms based on imaging and other techniques which could actually look at data in very high resolution, probably at the level of single cells or even finer. Now this opened up at least two types of challenges. One, of course, is maybe three challenges. One is data acquisition. Okay? So when you measure things at that high resolution, quality becomes a big challenge. To assure quality, to make sure that you are not looking at garbage is a big headache. So that was that. The second problem about such high resolution models is basically, are they reproducible results? Because there's a lot of stochastic elements to each individual cell's expression, and they can't be reproduced because they're not averages. They're not coming from a bulk which would kind of, where the signal would be preserved in some part of the bulk. So here, either a cell is giving you some signal or not, and you can't ask anybody else what that cell does. So that's another statistical challenge. The third was the challenge of heterogeneity. Okay, now that comes from molecular biology, where it's almost like somewhat in what you see in the era of atomic physics, back in the 30s and 40s. So you have to have statistics to actually tell you what's happening in the form of autonomous agents where cells are allowed to express themselves autonomously, individually. Okay, so how do you measure that heterogeneity? So these problems are some of these are classical nature in statistics. Like, for example, statisticians who do sample surveys and so on, they try to measure the health of an ecosystem or a forest by sampling parts of forests and ecosystems and try to get an idea of the heterogeneity, how to handle that. So we started looking at all those aspects, and that's again a decade's worth of research, nowhere near finished. So we are happy to collaborate. If you are interested, we'd be happy to pass on some of our publications and get views and feedback from you, and even collaborate if you want. So single cell analysis is going on, and probably it will also have some implications in the near future in clinical decision making and real life applications. Okay. Now the other dimension is in biostatistics and epidemiology area. And so I was telling you about the computational biosecurity team that I'm developing now. And this is partly funded by an NIH Fogarty grant. And so we look at traditional types of data like gene-environment interactions, applications in real life problems like nutrition, carcinogenesis, zoonosis, etc. But we are not just doing it in the usual way, which we are happy to do, but we also want to bring in this big data aspects to it. So in the Institute of Public Health, we have, since last year, started a health informatics MS and PhD program. And that health informatics program is basically geared to develop human resources for students and researchers for looking at different types of these large types of data. They could be satellite data, GIS data, all kinds of data that you could think of, from devices, from remote sensors, etc. So all these multi-stream data, sensing data, map data, distributed networks of sensors, etc. And then we try to put them together in predictive models and attach big data to biosecurity problems. So that's where the lab is, very distributed, and they're also geographically distributed. Makes it fun and challenge both at the same time. But if you have any questions, let me know. I'll be happy to. Get you in touch with the people who are working in each of these areas, okay? All right. So today's, in case you've forgotten by now what the title was, it was modeling the prevalence and the force of infection using serological survey data. This is from a standard textbook, not written by me. I'll give you the reference. It's basically a very recent textbook that has come out from Springer 2 years ago. So much of the material is sourced from there, and then I'll tell you how we fit in and how we are extending that work more in terms of what I present tomorrow. Okay. So the background is, so how do you measure the past exposure and the prevalence of some kind of infection that has occurred in the population? So there are several ways of probably answering this. One of the standard ways of doing this is to basically measure the expression of specific antibodies which are produced in plasma in response to antigens which are either a historic remnant of what's remaining from past infection or maybe induced by vaccination. Okay. So these antibody levels can be measured in blood serum samples using quantitative assays. So you get readouts with assays like ELISA, and there are various forms of ELISA which tells you, okay, so much antibody is present in a particular given sample, and there could be multivariate measures depending on the kind of panel of antibodies you're testing for. So per subject, you can measure generally half a dozen antibodies, but you could think of crunching this up using multiplexed assays and so on. So if you do have such data, then you could call it a serological survey data. If you do it in a longitudinal way, then you would have a surveillance system, and you could test then how much of from the antibody levels per subject, even if you don't follow the same subject, if you have it at population level. Let's say, for example, you're surveying dairy farms, okay, or you're looking for infections in chicken farms and things like that, most in the agricultural space. It's difficult to do such surveys in humans because it's not that easy. for a variety of reasons and good reasons. But they are done. They are done. And so serological surveys are conducted. They're often cross-sectional. So you basically do it once. But you could do it longitudinally, as I said. And they're conducted by epidemiologists to determine the prevalence of a certain disease in a population. Okay. How does such data look? So they typically look like this. So you have the antibody level, which here is depicted in log of that antibody level. So, and the y-axis tells you the frequency. So it's in the population. So you can see that if you have n subjects who are measured in the population. Yes? So that depends. So some of the so it depends on the way the immune system actually remembers things. And also, of course, the specificity of the test and the sensitivity of the test. And I'll tell you some of those issues which are always annoying. Depending on the kind of infection it is, I would say it's best for. So there's no general rule for some of the diseases. It would be best done within that year or so. And then there are some of these other kinds of things, like it's very age group dependent. So for example, neonates would have antibodies from the mother and so on. So that may differ from study to study. And the epidemiologists will have to take care of that as a part of the design issue. But if you are looking at antibody levels which are kind of produced as a result of vaccination, so then there will be surely a time component, and that's why they have booster doses and all that, right? So because the signal gets much weaker later on. So, but again, it depends on the type of infection. Some antibodies are produced kind of flat and all along, but that's rare. Generally, they decay with, and certainly with older people, the immune system is way weaker, and that. Right. Right. So the question is best understood in terms of the baselines that you have. So it's not just the measurement at a point of time that you're interested in, but if you had certain baselines of a given population, then that's. So tomorrow I'll touch upon some of these results. So without the right baselines, even if you measure antibodies, they are not going to be absolute. So you have to know how much change is meaningful relative to something else, something which is considered the normal immunophenotype. Okay. So this is basically a histogram. Okay. And it tells you that there are possibly 2 or if not more populations of people. And so for each subject, you've measured antibodies, and that's the antibody levels. And some antibodies are measured in high levels, some are in low levels, and therefore you can probably think that there is a susceptible population and probably there's another infected population. Susceptible populations are still not infected, and the infected population produces more antibody, and they were infected in the past, so they have higher levels. And there's always this intermediate population, which not many groups have looked into. We do study more on that. So the dynamics of this particular data set is what we look at, not a static picture. Most of the studies happen in the static space. So you do, because longitudinal study is hard to do, so you have one snapshot and you try to infer what you have to. Okay, so the same picture here, same data set, but the levels are shown here, and it's broken up, stratified by age of individuals, and then suddenly you probably see some interesting phenomena. But you can still see that this 2-class separation is still there, right? Fairly easy to separate this highly exposed, more antibody-producing subpopulation and the otherwise. Okay. So difficulty is, if you want to look at this classification and draw your conclusions, then the test, the diagnostic test, the measurement of antibody that you do, those kits and reagents that you buy from the manufacturer, they become kind of an important issue for both the health department as well as the statistician. The reason being they are not always the best cutoff for a variety of reasons. Some of these tests may not be very accurate or very generalizable. Some of these tests actually lack sensitivity, so they may not even have a good cutoff. But the worst is that something that you cannot change with your best technological development is the fact that populations are different. Okay, so for instance, So we have heard of tests not being effective for TB, which is a kind of which was big for several years, not very sensitive, not very specific. And then now, even in last month in India, there was a lot of issues about the dengue test, ELISA-based test again. So there are lots of these different companies which prescribe their tests, and then you go to the hospital, they run the tests, and they say, okay, maybe you have something, and then you never know whether to trust that or not, and then how to proceed with the therapy. So that all these poses their own challenge. So if you have to work with cutoffs, you're not very sure, so you could probably do it data-driven rather than cutoff-driven. Okay, so how do you determine the contours of an infection in terms of its past presence and its intermediate stages? So the standard way of going about this is you want to model the heterogeneity in the antibody levels. In that case, you assume that the sample is drawn from a population consisting of a generally unknown but finite number of subpopulations. If you, in mixture model terms, they are called components, resulting in a density function G that can be represented as a mixture of K density functions belonging to, let's say, the same family, to keep things simple. Tomorrow I'll show you more complex scenarios, and let's say each of those densities are called component densities, are denoted by F. Given a sample of antibody level yi, that's your data point for subject I, and let's say there's n subjects that you're measuring. The goal is to estimate the parameters of a K component mixture model. So it's a finite K, so it's a finite mixture model, which looks like this. So it's a weighted sum of density functions. Okay, so yi is your data, and that's your parameter vector, and there are K components, K equals 1 to capital K, and pi I are the mixing proportions. The sum of pi I, of course, pi K, of course, equal to 1. So there are K densities, which are basically, for example, there could be 2 densities here. So, and then depending on the choice of your density function, you could model them in a way which can tell you various things about what's happening on the tails, what's happening in the modes, and other kinds of inferences. Okay? So that's a general framework, very general framework. Okay. So now what you want to do, you want to classify the subpopulations of interest to facilitate targeted outbreak surveillance. So unlike the classical thresholding approach, finite mixture model, which we use, is a skew mixture models, allow identification of intermediate states of immunity that transition between the naive and or the susceptible populations and infected or fully immune classes of populations. So often you can see there's a lot of non-Gaussian, non-normal distributions. They're not very symmetric, and then they do have a lot of heavy tails, a lot of outliers or tail phenomena here. So we developed our family of distributions over the last 10 years. We have several papers showing which are the better distribution families. And we could actually then apply them and model what's happening in all these intermediate states where probably populations are losing immunity or gaining immunity, and there's a dynamics, there's a flux going on, and probably something interesting is happening there, because that's where phenomena start. Probably that's what we hypothesize, that if something is changing, then probably it's changing more at the tails rather than in the center. Okay, or something is evolving in the form of new kinds of resistance, a new kind of vulnerability rising in the population. Probably they happen more often in the tails. That's a hypothesis, of course, but you can test them in some extent. Okay. In addition, longitudinal serological surveys allow temporal patterns for surveillance. That's also what we are trying to do, especially if conducted in populations that reside in and around natural reservoirs of infectious agents. So we know that.
Pyne, just again the question. Yeah. So this is the classification problem.
Yeah.
Where you have a bunch of components.
Right.
Each governed by an individual probability distribution.
Right.
And you want to ask if a particular sample, you have a bunch of samples, and you want to ask where do they fit into this.
That's right. So, and that plus the fact that if you want to do it as a surveillance, then you have to assume that these populations are fluid, in a sense they're changing all the time. Not that it's always easy to collect such data, but let's say you want to create a framework for that, then you also need the math to be right so that it can capture those changes in the parameters, and they have those parameters which are sensitive to those changes. Otherwise, you can have a nicely fitted model, but it would be a rigid fit, and there won't be parameters which can quickly capture those changes. So that's where our research lies, but I'll go to that, more of that tomorrow. I'll now tell you the general framework today. Okay, the assumptions are an individual in a population is either in the infected state or in the susceptible state. For today, it's more of a rigid structure. Okay, the disease is in a steady state. That's the second assumption. That is, the transmission parameters which you estimate, want to estimate, are time independent. Okay, the disease is irreversible. That is, the immunity is assumed to be lifelong. So all of these are kind of limited assumptions. They're generally not true in general. Okay. And the other assumption is that the infected persons are alive, generally alive. And the final assumption is that the above assumption could be really unrealistic, okay, in the real life. But that's to get the mathematics going, we need to make these assumptions. And so let's go ahead with that. Okay, so some terms and definitions. The prevalence of infection and the force of infection are important parameters, so we need some dedicated notation. Prevalence is generally denoted by pi, is the proportion of infected individuals at a specific time in a certain population. The force of infection lambda is the instantaneous per capita per person rate at which a susceptible individual acquires infection. Okay. These are very standard definitions, so you'll find them in any textbook. This matter is, as I said, derived mostly from a textbook. So the basic approach is that the prevalence and the force of infection are commonly estimated using the current status data from a cross-sectional survey. So you basically just have static data, okay, from a cross-sectional survey. And there has been several such papers in the past showing that these type of approaches might be useful. It is assumed that the current infection status, Zi, of a subject I, so this is the classification coming in, okay, aged AI, this is the age of subject I, is known, and let's say it has a Bernoulli distribution, which means it's a class label 0 or 1 with the probability pi AI of being infected in age AI for subject I, and 1 minus that probability if he or she is susceptible. Okay, so here pi AI, as I told you, is the probability to be infected by age AI. The variable Zi, which is the class label, is obtained by dichotomizing the individual antibody levels. So Yi is known to you, that is your data point for the ith person. You use a cutoff point phi, and so the prevalence is given by pi AI, is the probability of this Zi, this class label being 1 given AI, the age, and that is determined by the cutoff. If the amount of the antibody level is Yi is greater than phi or not. If it is greater than phi, then it's 1, otherwise it's 0. Okay, simple dichotomy. So there are different ways to model pi AI in terms of age, from parametric models to semi-parametric models to non-parametric models. So this is, again, as I said, decades worth of research. Our work is more in the parametric space because we love to do it that way, but there's no need to be dogmatic about it. There are lots of very good research in any of these approaches. But all of them try to maximize the likelihood, the log likelihood function, as a function of the model parameters, the model that you're trying to fit, given the data, based on the data points. The data is, of course, here, Zi and AI, and the Zi is defined in terms of this threshold phi, okay, for n subjects. And this is the log likelihood function. Okay, so that's how the data looks. The text is from the figures are from the textbook. Okay, so again, this is your log likelihood. Log of antibody levels. That's your log of Yi. So the data looks like this. There are classes. This is the same data shown as histogram. You can see that most of the times these type of surveys give you these skewed and heavy tail distributions. So I don't know why it's not popular. Some groups use our distributions now in Austria and Germany and some other groups. But we were the ones who started looking at them as non-Gaussian distributions initially. And then you can dichotomize them, the same data, in 0 and 1 spectrum, and two ends of the spectrum, and that's how it looks. And if you want to kind of represent them as a function of the number of people who are in that age group, then you can use other graphical tools like big dots and small dots, which size of the dot is proportional to the age group it belongs. So it depends on the number of people in that age group that you have. So this is how the seroprevalence looks, pi AI. Okay. So you can see typically as a function of age, initially few people have the disease, most of them are susceptible. when they're young and they produce less amount of antibodies. As time goes on, sooner or later they get infected, then they have this memory of that infection and more antibodies keep getting produced, and finally everybody is infected and then it stabilizes, right? So it's close to 1. So almost 100% of the people have the that particular antibody level that they reach a saturation point. Okay, so now again the math framework, back to the framework, very basic framework. I'll just take you through it, and if you have any questions, you can either ask me or I can refer you to the textbook. So how do you model force of infection in an age-dependent manner? So in generalized linear models, age-dependent probability of infection pi a at a age a is modeled as, we have seen this function before, but now you have this link function g. So g is an arbitrary function. It's in GLM terminology, it's called a link function of probability of z equals 1. You remember, we saw this class label before. Given age a is written as g of pi a. We have defined this before. And this can be written in the form of a link predictor called eta a. So here pi a is modeled through a so-called link function g, and eta a is a so-called link predictor. So this is standard terminology for GLMs. If you're aware of that, you would probably recognize it readily. So using a so-called catalytic model with log link, this is a very standard textbook approach. That is, if you take pi a equals 1 minus e to the minus eta a. So if you take log of both sides, you will get this back. Okay. So we get eta a to be a cumulative hazard, and hence force of infection lambda a is the hazard function here, is simply the first derivative of the linear predictor. So the linear predictor is eta a. So lambda a is just the first derivative of eta a, and pi a, the prevalence, is 1 minus e to the minus eta a. So once you know the data, you actually can estimate them directly from the serological data, both your pi a and lambda a, force of infection and prevalence. Okay, so just plug in the values and they will give you. Okay, so now to plug in the values, of course you have to estimate the parameters from the data first, and there are many of those modeling approaches. So you fit a model and you estimate the parameters, and so I'll show you today some parametric approaches, but as I said, there are other approaches which are probably equally as good. So this is how data looks like from the textbook. These are in certain European populations. They have measured things like measles antibody and so on. So you have seroprevalence on the y-axis, you have x-axis as age, and you can see the curve gives you the fit of the data. The data real points are these points here. The size of the points tell you how many people are there in that particular age group, and then you can also see the corresponding force of infection, lambda a, is shown here. Okay. Similarly, if you want to use a variety of other types of modeling strategies, you would get other kinds of fit. So they have different shapes, but inferences drawn will vary a bit from one model to another, but that's the basic idea. Okay. So note there's something funny here. You can see that although we know that this curve is supposed to kind of go up and up and up, there could be certain times which are funny, like there is a dip here and there is a kind of tick up there. Any guess why it might be? So why would this be? So here initially it's showing that there is some seroprevalence. The serum has more antibodies initially than, it should be kind of not having much antibodies at all. It should start from kind of a clean slate, but it doesn't. Any guess why that might be? I've already probably told you before that's because of the child actually during birth inheriting some of the antibody. Inheriting is not the right word. Getting some of the antibodies from the mom. Okay, and that's also a very active area of research of what type of antibodies you get and when you don't get and so on. And then there could be other kinds of immunologic exposure issues, like the way birth is given, the way in situ exposures, in utero exposures are, and so on. So that's another very active field of research on innate immunity, which we won't get into here. So there could be antibody levels which are kind of acquired in a neonatal stage, which you lose, and then you kind of keep getting, and then sometimes funny things happen, either due to modeling or due to measurement or due to things that we don't know. And then there could be some lower prevalence, but prevalence in general keeps increasing as it keeps going towards 100%. So finally, the entire population is exposed, and they're producing the antibody. Okay, so we said that I said that we use mixture models. I already told you what they are. There are various ways of going about doing this, and I'll tell you one way that we do now and another way that we do tomorrow. But they're all basically just computational tricks or ways of getting things done. Okay, so as you know, knowledge of the antibody level, threshold level alpha phi. So I told you we use antibody threshold level phi to classify the population as who has the right amount and who has less than that, right? So assume no such knowledge. So let the data tell you. And so we'll model the data and try to infer a classification. I mean, not so discretely, but from the parameters of the mixing proportions of the two classes. Okay. Hence, we use a mixture model on the serological survey data. I showed you the type of data. The mixture model parameters can be estimated, as I told you, using many algorithmic tricks and approaches. So, for example, maximum likelihood methods, expectation maximization type methods, which I'll show you tomorrow. That's what we use most. But we also use usual MCMC methods based on Gibbs sampling and other techniques, which the students might do, especially those who are computationally oriented, may be interested in. And we also use Bayesian techniques for reasons where Bayesian techniques are better or better suited. For parameters via the posterior simulation is possible after you do MCMC and fast computing. So a lot of our work on fast computing is basically to support our Bayesian inference techniques. For mixture models, in using Bayesian methods, the priors must be chosen carefully. So if you're interested in the mathematical aspect of it, I'd be very happy to discuss that with you, maybe after the talk. Often we use hierarchical mixture models for that purpose, where not only the parameters are estimated, but they're assumed to actually are drawn from distributions. So those distributions parameters themselves come from higher level distributions, defined as hyperparameters, which actually is a reason why we call them hierarchical model, and we have techniques to fit them in an efficient manner. So after you fit them, how much time do I have? Okay, because I can actually make this kind of continuous talk going into tomorrow. So it's okay if I end abruptly, but I hope so. So these are different populations, and you want to kind of estimate the mu1, mu2 are the means of these distributions. But more than that, you're also interested in making the inference. You're not assuming any knowledge of phi, the threshold which actually classifies them. So you're letting the data tell you d, which is the difference between the two populations. And so you need a parameterization for d as well. Okay. And so typically data looks like this, and this is when you fit a normal mixture. So you have two normals, and then probably there is some separation, natural separation there, but often you cannot assure that, especially if you are interested in dynamics of populations changing their mean location. So that's something which not every team looks at, but we are interested in that. So if you're interested in that, it might be interesting to have these parameters chosen for capturing these differences and so on, which are flexible. Okay, hierarchical Bayesian mixture models via age-independent prevalence. In order to model the heterogeneity in the antibody levels, we assume that the sample is drawn from, I've already gone through this. I don't know why this slide is repeated. Okay, so here FYI, given the parameters are the component densities, where pi k, as you know, are the mixing proportions, component weights, such that the sum of pi k is 1. So these are the component weights, because it's a finite mixture, so it's a finite sum. Pi 1 to pi k, the sum is 1. And then the psi k are the vectors of density parameters which have to be estimated. So these are the parameters of each component's density function. Okay, In particular, we focus on a hierarchical Bayesian mixture model of 2 components in this case, if you assume just 2 subpopulations, with possibly different mean and variance parameters, such that the first component models the distribution of the antibody levels of the susceptible subpopulation, and the second component corresponding to the infected population. And so because infected population produces higher levels of antibodies, so I'm calling it second, basically means the one which has higher mean. Yeah, so that's exactly what it is. We showed you before that the prevalence changes with age. Yeah. And you could do it in an age-dependent manner also. Okay. So you can stratify the data first and then do the model. And the mixing proportion is pi. So you have 2 density functions. They're mixing with each other with proportion pi is to 1 minus pi. So, and the data is given to you, so you can actually basically model this function. How do you do it? You introduce a latent variable. As I said, it's a classifier variable, which is an indicator variable, 0, 1, which represents the individual i's unknown infection. This is a latent variable. It's not told to you. You don't know it. You will have to understand its status given the data as part of the modeling. And so it's unknown infection status, assumed to, let's say, come from a coin toss distribution, Bernoulli distribution. Again, the parameter is pi. Thus, the densities also gives you the classification straight away. So you know how to label it. It's like either z equals 0 or z equals 1. So one of these components survives, the other one is 0. Okay. Importantly, the mixing proportion parameter pi is the probability that an individual. So this is an interesting observation. So you did your modeling. You found the mixing proportion pi. So that pi is in terms of the data that you have. Okay. Now, that mixing proportion pi is the probability that an individual in the population belongs to the infected component. So that's the prevalence part, seroprevalence part, and thus it can be interpreted as the prevalence of infection in the population. So in other words, you are trying to estimate prevalence, the amount of, remember I defined prevalence in the earlier slide, the probability that a person is falling sick and the prevalence. So now that you're trying to estimate directly from serological data from yi. Okay? So yi is your input, but it is input in terms of serology, in terms of your antibody levels. So you're you're taking seroprevalence and calling it prevalence. So this is a direct measurement. You do not have do not need to do anything else. Directly from data, which is antibody level data, you're estimating you're fitting the mixture model. You have your parameter pi, and that pi you're calling your prevalence. So that pi is actually seroprevalence. It's basically amount of antibody levels difference between the 2 subpopulations. Pi is to 1 minus pi. And now you're calling that as prevalence. So you're directly measuring pi out of serology data. And once you know how to do pi, you can go to lambda. Yeah.
Yeah, so you mentioned that we could have made this age structured.
Right.
So suppose we had done an age specific pi. So let's say we index pi with some
right.
age
We actually saw some pictures also.
Yeah, right. So would you expect pi I to monotonically change with I?
So let me show you the pictures. So that's seroprevalence. Okay.
Right.
So that's changing over age. Okay.
Right.
So these points are real data, of course, because they're intervals. You have a way to weigh them.
Right.
And this is the fitted model.
Yeah.
And then there is the fitted model, and then you show it as a classification that there are 2 subpopulations that you can do. That's for overall for the whole population, but you can surely see that there is a trend here. It's supposed to be monotonic. As I said, sometimes funny things happen, which could be either something about measurement, something about something else too, biologically meaningful too. Like why would prevalence go down? So you're always supposed to get infected. You don't get de-infected or something like that, but you never know. There are certain times when you can actually reduce, let's say during, for example, cancer patients and so on. So they could have weakened immune system for a variety of reasons. But that's not, this is population data. This is not such special cases. So we don't really know why would prevalence go down. Okay, this is seroprevalence. This is the amount of antibody measured. Okay.
I thought you mentioned this was measles, right?
I don't remember. I should have probably tagged that because this was a classroom lecture, so everybody had the book. I didn't bother, but I could tell you that, which it is for. But so sometimes this type of funny things happen, but you could also reason that probably this is not real. It is basically, if you fit the model using some other model, maybe this dip would vary and go away. Okay, but we don't know. So I don't think people get suddenly, for some reason, they get acquire. I wouldn't have any sane explanation, but I'm very sure immunologists would have some insight of why this could happen. I don't know, maybe at what age. So this is mid-20s.
No, I mean,
I don't know.
I'm just thinking
Why
of this
would-
from a very intuitive sense.
No,
like,
sometimes you can. Okay, go on. Yeah.
just suppose you start with, essentially a two-population model.
Right.
Initially, at very early ages, the bulk of the population would be in the susceptible, and very few in the exposed.
Right.
And gradually, as age develops, more and more of the population is going to go from the low peak to the high peak.
That's
so
right.
you expect, at least for measles,
Right.
something
So
like a
for most of
monotonic
them,
curve.
it's almost. I mean, it's very common. You never see a very deviant pattern from this one.
Right.
So this one probably I chose because it had that dip, but most of them wouldn't have that dip. You wouldn't suddenly stop producing antibody. I don't know, unless something very funny happens to a population. That could be another kind of exposure, or some data which was sampled differently or something like that, part of design of the way the sample was done. So, but generally they're very predictable. They just go up and up and up. That's it. So remember that in certain other subgroups, it's even difficult to collect data. So data quality becomes another issue for that reason. Okay. So now, as I said, that we could assume that seroprevalence could be assumed to be prevalence, and this was a well-known paper which actually brought this point home, and then we live with that. Okay, so for students, if you are interested, you could do it as a starting exercise as mixture of two Gaussians. There are several ways, as I told you, how to fit such mixture models. So mixture of two Gaussians is not a bivariate Gaussian as such. It's basically a weighted sum of two univariate Gaussians. Of course, you could do it in multivariate space too, but as a starting exercise, you could assume that this is univariate data. You're just measuring one antibody per subject, and so it's basically you take two normal distributions and mix them in proportion pi 1 and pi 2. Pi and 1 minus pi, and then you need to estimate the model parameters, which is the mean and standard deviation of each distribution, each density function, and mu and sigma, and the mixing parameter pi. The seroprevalence parameter can be interpreted as the prevalence, as I told you, thus connecting the infection with serology. Okay? So the infection is a population parameter. Serology is a very specific parameter, need not be population-centric, but you're measuring serology, so you live with that. Assume that the mixture probability pi is constant across different age groups, as I told you, and that it follows a uniform distribution. So here the pi itself could be a function, could be a parameter which could be changing, which could be drawn from a distribution. So it could be a random variable on its own, right? Which means over age, the mixing of the subpopulation, if you stratify them by age, that itself could keep changing. Okay. So in order to then make the model flexible, you have to have these parameters, have to have them adequately parameterized so that you can estimate them. Of course, time, complexity, and other headaches will go up, but that's part of the game. We assume that in a sensitive assay, the larger values of the antibody levels indicate a seropositive status, which means if the assay is sensitive, then the two populations will be well separated. I mean, there will be, there won't be two overlapping subpopulations. There is significant difference between the two. Okay, and therefore we include that, or we include an order restriction, which means the means will be adequately separated, means of two subpopulations in the model. Therefore, we assume a constrained non-informative prior for mu1. Mu1 is the mean of the first population, which is at most up to mu2, because there's a difference between mu1 and mu2. So you can let this parameter come from uniform 0 to mu2. Mu2 is separated from mu1 by an amount delta or d, which itself can be coming from an uniform distribution, 0C, where C is a relatively high constant, which means you allow space flexibility for the populations to move around. The parameter delta is not fixed, but represents the shift of the mean antibody level between the two components of the mixture. Yes?
You're actually taking the log of the antibody level.
Yeah, I think so. Sorry. So I should have been more careful about that, because we almost always say antibody levels and almost always mean log of antibody levels. Okay. To complete the specification of the hierarchical model, we assume the following non-informative hyperprior distribution. This is for the precision parameter, so 1 by sigma. So what is sigma square in this case? 1 by sigma square, it comes from a gamma distribution. Here gamma alpha beta, this are the alpha beta values, denotes, you can parameterize even these, but let's say we stop here and we have only one level of hyperparameters, denote a gamma distribution with mean equal to alpha by, this is just gamma distributions properties, this is mean and that's variance. Hence by choosing alpha and beta as very small, the prior distribution for both precision parameters, sigma 1 by sigma is a gamma distribution with mean 1. You can see these two are equal values, and large variance because beta square will be much smaller than alpha, which reflects our uncertainty about the true values of the parameters. In other words, we don't know the parameters, and we are kind of being honest about it. We are letting our uncertainty be parameterized this way. Okay. So the model fitting, again, as I told you, is basically you want to find the mu1 and mu2 estimates for that. And then after you do that, you also want to, along with that, you also want to find estimate for delta, which is the difference between the two means. Okay. You could do it in an age-dependent fashion, and I'll wrap up. I have exactly 1 minute to go. So probably it's just the same methodology. Okay, you could, instead of doing it as pi a, you could do it as pi a. Instead of doing it as pi, you could do it as pi a. Pi a is age-dependent. Okay. The only thing that you have to remember is that you have to make the model a little more complicated, but that's not by much. And so, for example, you can take a log logistic model where you get this logistic function here as part of a logarithmic terms in terms of age. Okay. So if you fit this model, you can estimate prevalence again, but in an age-dependent fashion. Okay, pi, but in terms of a, and that's about it. And then, as if you know pi, you can also find out what's lambda, force of infection. And then you could do this using simulation. As I said, that this is one of the methods of estimating parameters. You could fit the model using simulations, and then basically get a good, estimate of the posteriors of the parameters, of the distribution of parameters you're looking at. You could do MCMC methods in R using a lot of different packages. If you're interested, just let me know. Some packages are better than others in terms of convergence rates and so on. So that's basically all for today. I'll just give you the classical inspiration for that, which I found very interesting. So our lab has been working on finite mixture models for like 10, 11 years. But most the classical history of that goes back to 1890s. Pearson was trying to fit a mixture of two univariate Gaussians to data on Naples crabs using the method of moments, and that's another way of doing things to distinguish the divergence of species. So this is, I'm just telling you to the students of how this interesting field came about. This is one of the early studies. Carl Pearson was trying to fit data from, let's say, the size or some dimension of Naples crabs that he measured, and he wanted to test whether there are not just, it's a unimodal distribution, there's only one peak, but probably there are two species, one subspecies splitting out and diverging from another. So in order to show that, he manages to give a nice fit using two Gaussians, and he's showing that how one species is probably having bigger or smaller size, and you can see in the tails it's kind of moving out as splitting into two distributions, which is not otherwise easy to see in terms of the histogram, the blue data. So that's basically the idea that we got inspired by, that can we capture these diverging dynamics, not just in static data, but if something is becoming different, and you could apply this in genetics, you could apply this in evolution, you could apply this in ecology studies. If something is slowly and or even sometimes within smaller time scales changing and becoming different, can you capture that using parametric models? Okay. So I'll show you more on these stuff tomorrow, but I just skip everything. So I'll show you all these different skew algorithms that we developed. And that's all for now. If you have any questions, I'm here full day today, tomorrow. You can literally, get hold of me. So thanks to everybody for your attention. Thanks to the funding agencies. These are my email addresses. If you're interested, just shoot a mail. I'm not that bad a person in replying. Sometimes it gets late when I'm traveling, but otherwise I do. And just in case you're interested, this is the earliest known representation I could find of an infectious disease. It's in a Egyptian relief. You can see that this particular person has probably an early polio infection. So this is from 1400 BC. So that's all. Thank you for the attention. If you have any questions, feel free. Any questions? No?
What is the textbook that you were mentioning?
That's what I was trying to actually include that slide. Okay, so I'll give you I know the textbook. I need to remember the author's name. Okay, so I got stuck with that. I'll get back tomorrow. Okay, so I use that for my teaching. So, thanks. No, that's a textbook on doing this type of estimation.
On seroprevalence.
On seroprevalence or this type of data. There's other elements also they have on contact mixing and other stuff, but I'll give you the reference. I actually thought it's a shorter talk, so I took out some of the slides, and now I'll bring them back.
All right, so if there are no further questions, let's thank the speaker again, and we move on to the next talk.